First, I would like to thank the organizers to invite me to present my work. And this is the idea that the way that I think is like I work with transgenic mosquitoes, but also I think the transgenic mosquitoes or other any technology for male release need to be part of the integrate control. And this is the way that I see the integrate control. And for sure, the education of the community engagement is in the middle because it's the best part that we really need to improve and talk with the community and explain all this new technology that we are trying to use to control the mosquitoes. So my talk is going to be on population suppression, and of course I'm not going to talk about the regular SIT and the IIT because people are talking. I am going to talk about transgenics. So the way that I see, we can control mosquitoes, and the idea is if you use transgenic technology, you can have a transgenic line to do the suppression, but also what's happened, we are always using, having the residue mosquitoes. It's very difficult to eradicate the mosquitoes. It's almost impossible. So this idea is if you have this a small number of mosquitoes, those are the mosquitoes that still transmit the disease, okay? So then the second approach that we need to have is replacement. And these mosquitoes now is fighting the virus, not the mosquitoes, okay? They are going to be the vehicle for fighting the virus transmission, the blocking transmission. So first I'm going to talk about the suppression, transgenic lines, and of course, I said that before, I was involved in the evaluation for the Oxitec transgenic line, the riddle technology, for 5 years. We released at least 3 different pilots for the trial. And what we learned on this approach, and we produced the mass rearing protocols and facility, and we showed that we can do this. It's not that terrible that the people think that is very terrible. We can produce at least the last program that we have, we produce 1.5 million per week for the males to release, and we use this facility to develop transportation system for the production facility to the place that we are going to release the mosquitoes. We have some protocols for that, and a lot of countries are learning this kind of mass production and transportation technology with us. That is very nice. Of course, each country adjusts this mass production. This is the idea, to have better and better protocols. This was the first one, but we can do it. And we can show that we can suppress the population very, very fast. Depends how much you are going to release the mosquitoes. And if you use those mosquitoes together with the other methodologies for the integrate control, you can reach the same amount of reduction using less mosquitoes to release, and the technology is going to be less and less expensive, okay? So, what we learn about that? How can we implement the transgenic technology in a country? So, the idea is to have the egg distribution instead to have the pupa distribution, because it's much easier. So, to have the egg distribution, we need to have a different kind of thinking. So the products for transgenic technology need to be no larva sorter. We don't need to have females and males for production. Imagine that you have a biofactory that 70% of your production is trash. This is not good. This is why it's so expensive. So if we have a line of mosquitoes that everybody's going to be male, this is the best production that we have. So this is the technology that we are trying to find it, or the product that we are trying to produce. So the tetracycline needs for today, for the Oxitec technology, the egg production and the male production, everybody needs to be raising tetracycline. The idea is to have a product that is less tetracycline, that can be less chemical to use and less expensive. So, and the idea is what's happened today, this riddle technology, the larvae die, larva die. They die between larva 4 to pupa. So what's happened, the health agents go to the houses and see the larvae. And they know that maybe can be transgenic, but this is a positive house. This is not good. And then they ask me if we can produce a mosquito that is no offspring, that it can be sterile. So this was the point that we have. So we came with this idea to produce a genetic testing strain that is going to be male-only production, sterile male, and then the tetracycline is going to be used only in the colonies because both genes are going to be shut down just for egg production, not for the male production, okay? So we came with this idea, this sterilely conditional construct, is no antidote, then you have a male and females, is no viable eggs. If you have tetracycline, you have a viable eggs. We produce this construct. Here is just like the example for 2 constructs that we did, and we use an endonuclease to destroy the DNA, is one kind of concept, and another is for apoptosis activation, and then you can kill the spermatozoid, and everything is going to happen inside the spermatozoid only. And we did the first construct, and then we have like these transgenic lines, and we have a very good one that is almost homozygous, and we have a very nice reduction for the larvae hatching. One thing that I would like to say, all this technology is producing inside the university, and this is going to be free. At each country that want to apply these mosquitoes is going to receive the mosquitoes for free, okay? There's no profit in this. So we produce a sterile male, this is going to work, and we are producing the GSS strain. The idea for the GSS strain is 2 thinkings, and not only me that are working on this kind of production, the construct, but few labs. And the idea is or to kill the females in the embryos stage, or just make you break the pathway for the double sex, and then all the females are going to be males, okay? This is the 2 ideas that we are having. We have some transgenic lines that is being produced, but we still don't have the results, and maybe we can have this for very soon, okay? So this was the idea why I say that we have the second step. This was the Singapore 35 years of information, and what you can see less mosquitoes, but you still can have the disease transmission. And this is why we came with this idea that we need to have a fight for the virus. So my idea of fighting this virus is a suicidal model. What is that? We learned that even if you have reduced the virus or the parasite inside the mosquito 99%, you still can have the transmission of the disease. We use these models as a malaria model, and for mice malaria, for avian malaria, we show that even if you really reduce the parasite, we still can have the transmission of the disease. It's less transmission, but it is still there, okay? So we came with this idea. If the mosquitoes know when they feed on infective blood, they know they are infected. So can we use this information to trigger a gene and kill this mosquito? This was the first thinking, okay? So the idea is you have the natural population, and then you have a transgenic line that is going to introduce the gene in this population. This is going to be spread when you have the mosquitoes that is be infected by the virus. These mosquitoes are being killed. And the idea is blocking completely the transmission of the virus, okay? So This is a lot of biochemistry, but this is the idea how my gene works. All the cells are producing this gene, and because you have the polyprotein in the system for the dengue, and the cleavage sites for specific for dengue polyprotein, this is a dengue proof of concept. We put this signal here in our protein, and because everything works in the ER, we put a fusion protein that is a ER localization and apoptosis little protein, okay? So the idea is if you have all the mosquitoes, all cells of the mosquitoes, all the populations are producing the genes, the protein is there. If you have a dengue infection, you can process the protein, and then you have dual system. One is in the midgut, that you can kill the virus replication or kill the mosquitoes if it's this dissemination step. Okay? No. Too much? Okay, but the idea is kill the mosquito. Okay? This is the idea. Okay? So we did the transgenic lines, and we showed that some are working, not totally the way that we think that is working. We got like around 60% of mortality. We want to have 100% of mortality. So we are adjusting our gene. We are changing the Michelob for a different kind of protein that can be stronger, the signal. And we just got the constructs, and we are trying to have the transgenic line. The idea is we are going to show that we can have for dengue, Zika, and chikungunya in separate lines, but then we can put all the sequence together as a target, and then produce mosquitoes that can be used for the flavorvirus or arbovirus in general, okay? Because all these proteins are very conservative, okay? This is the idea, and I would like to thank the people from University of Sao Paulo, that is the people that produce the transgenic lines, and the Mosca Med Brazil, that was the people that work in the facility to produce thousands and thousands and millions of mosquitoes and eggs. Thank you very much. That's it.